All right, testing. There we go. Okay, sorry about that. For those who couldn't hear me a moment ago, thank you for letting me know. And for those of you who are just joining, real quick, we are a little behind on the syllabus. I'd like to give you more than one lecture on the BOD stuff. So I'm going to try to condense this one here, start on that for the next time. And worst case scenario, we might shift and do an alternate exam review and give you a little more time on that homework. Okay, sound good now, I hope. And back to the secondary treatment with recycle here. Last time we talked about no recycle systems, really looking at the mass balance of how microbes are growing. So in a recycle system, we've got lots of bacteria. That's going to change our mass balances. And in fact, we're not going to do mass balances for this portion of it per se, or exactly how we used to. Instead, it's going to be, we're going to be dealing with a system where we have some known relationships between how much mass exists in the chamber, how much we're wasting, things like that. So it's not going to feel like the same type of mass balance. It's still principled on mass balances. And if you look at the overall system here, we have a boundary here, a boundary here, and a boundary here. If we draw our control volume like that, we have X naught is 0 there. XE should be, if the clarifier is working, 0 here. So then this X waste or recycle, I guess it's labeled as recycle, it's the same amount, right? Same difference. We do have an X there, and that's because we are growing X inside, right? If X is increasing inside, and yet we keep the entire system at steady state, well, we have to be removing X somewhere, right? So when we start dealing with a recycle system, what we'll find is we have several equations to relate to one another. And we'll also see that this, the amount that we are sending out the waste line happens to be very important and can control a lot of what we've got going on inside. Okay, so those little things I just sketched up are kind of our assumptions. First of all, we assume that the clarifier is working and working well enough to settle basically all of the bacterial mass, anything we could have been able to weigh, right? Certainly some bacteria are free-floating and escaping, but not in large enough quantities to really weigh and see that difference. We're also going to assume that there's no biological activity in the clarifier. That's because we're not bubbling it with air. If we were bubbling it with air, we'd have a hard time settling anything, right? It's going to be mixing it, it's going to be floating things, it's going to be bad news. So we can't use that volume, so it's as if the volume is 0. If there's no biological activity, whenever we do, our mass balance, and we say V times some reaction, let's say R prime of G, if there's no reaction going in there because there's no oxygen being added, we just can say the volume goes to 0 or the reaction goes to 0 in there, whatever, however you want to say it. It's going to be discounted as if that it doesn't matter at all. Okay, so then the other thing we can notice is our substrate going through the system. The clarifier doesn't change anything about that. The only thing that's changing the substrate is the bacteria eating it in the tank. Okay, so any substrate after that, that's just what it is, right? So the amount of substrate here is the same amount here, the same amount here, here, and here. That's all I'm saying with that next assumption. That's pretty obvious if you think about it. And the amount of volume or the amount of flow rate we recycle here, technically it will dilute the substrate a little bit. It's incoming, but it's generally not a huge flow rate. So we could take a look at a problem like that, but it's not a huge flow rate. And last, we noticed that XR in the return line here should always be above what's in here because we've concentrated it. XE should be 0. XR should be larger than what we had in our tank. And XR is the same thing as it's leaving out the waste. Okay, so there's our assumptions that we can use to get a bit of a handle on what we're doing with the solids and how we're managing the system. So overall, what we are really aiming to do is in some way manage that growth, right? Last time we talked about the rate constants that were dependent on how much food the bacteria had. One of the most important features of this is what we would call the food to mass ratio. So F/M ratio. This is one term, and we're going to leave it, and we're just using the book's terminology. We're going to use it in this equation here that's given. And I want you to know that this is not food and mass. This is not two different variables. So if you see F/M, keep it as F/M. Treat that as one variable. Okay, don't multiply both sides by m. Don't do anything like that. Just leave it F/M. It's just one variable. I mean, technically, if you needed to break it up into parts, we're going to use the equation for that, because on the top here we have food, and on the bottom we have the mass of the microbes. But we're actually, it's not just the F/M ratio as just a strict ratio, it's a daily ratio. Like how much food per day is some mass of microbes obtaining. So even in that, the term F/M is a little perhaps confusing. Okay, but this is, if you recall, based on our analysis of at what point are the microbes, really happy to grow and growing the fastest they can. Well, you might imagine that the way to find that is this food to mass ratio, right? If it's a very high food to mass ratio, they're probably growing at their maximum specific growth rate. If not, there's limited growth there. So that's this reminder here. This mu depends on that amount of food. And so when we control a system like this, one of the important parts is, well, how much food are we giving them? We can't actually control how much food we're giving them because we're not going to tell people to stop flushing or flush more or whatever. We can't control what's coming down to the wastewater treatment plant, but we can control the amount of mass of microbes. So here's one of these points where we see that we can control something that's important to the process to optimize it, to keep it growing well. So let's take a look at the formula real quick. I already mentioned this, but on top, this is a flow rate times the substrate. Substrate's the amount of food coming in, that's not. The Q, that's the flow rate. That's essentially, if we take a volume per time, that's the flow rate, multiplied by a concentration, a mass per volume, the volumes will cancel, you'll get a mass per time, right? So that's the food, the daily food given. And on the bottom, it's the same thing, except we have the volume instead of the flow rate. And so that's just, okay, in that volume times the concentration of microbes, that's the mass of microbes, right? So it's very simply mass per time kind of a thing. So that's milligrams BOD per milligrams VSS per day. So if we get a handle on that, we can't change the volume, we can't change the flow rate typically. Again, can't tell people to stop flushing. So what we can change is the amount of bacteria we have present by removing more or less of them. And it turns out this F/M is pretty critical for making sure we have the right microbes growing. If we get a very bad ratio, or maybe there's some weird other food type, so we'd prefer it to be a consistent food type. We would hope that we don't have a sudden spike in pH one way or the other, or sudden, normally we get sewage and then suddenly we're getting a whole bunch of oil and grease from restaurants or something, who knows what. If something like that would happen, there's potential for different types of bacteria to start growing and you can get some that like to form more filamentous, floaty type stuff, and then you have a whole bunch of mass escaping, and that's bad news. Right? I think we talked about that before, but basically just don't ever want that. So part of controlling the food to mass ratio is just to ensure we are consistently getting the same types of microbes in approximately the same amount, so we have the same treatment and a consistent treatment with an effective clarifier. So all of those are quite important there. Okay, so how do we control the amount of food there or the amount of bacteria there? Well, we can introduce something on the next slide, actually, for the solids retention time. But first, let's just, since I have this slide first, let's just note here that our flow rate, our hydraulic retention time is not actually going to change even with the recycle. So hydraulic retention is V over Q. And if you recognize we're drawing our control boundary all the way outside of the clarifier, but we said the volume of the clarifier is effectively 0 for not having any biological growth, then the volume that matters is actually just the aeration tank. So the volume that matters for reactions, right? That's how you might want to look at it, is the hydraulic retention time for the reactor itself that matters. That's going to be the volume of this tank. So with that, even though we are recycling some water in there, the volume didn't change and the total flow rate into the system and out of the system doesn't change, right? There's an internal circulation thing, but across the whole system, we still have the same flow. So Q is going to be equal to QE plus QW. So that's not, the QR there, it doesn't matter how fast you recycle, how much you recycle, the overall retention time is going to be the same. Okay. Now, we do have control, and we can change something called solids retention time. Now, some people also call this cell retention time, and so that's where we get a theta C. So theta for retention time and C for the cells inside, the bacteria cells. This is a description for the amount of time that the mass of bacteria stay in our system. And that will be changed according to how we manage our waste line versus recycle. So we can see in this picture, I think this is a solids return line here. A nice gooey sludge just to recharge the system with a good bit of bacteria to keep them going and preventing over dilution. Okay, so then if we had no recycle, the solids retention time should be the same as the liquid retention time, right? The hydraulic, because it's just the flow rate is taking the solids through, and that's that, right? So if in terms of the amount of time that it's spending in there, the theta C would be equal to theta if there was no recycle. But since we're recycling, we have to ask ourselves, well, how do we get a value of this? And if theta is V over Q, we imagine that that theta C should be something like V times X over Q times X. Something of that form, right? But then we recognize that we have a couple different flow rates leaving, so we actually have to account for that. So when we take a look, we'll say theta C is V times X, right? It's that stuff that's there, divided by so it's a V over Q form, but with X accordingly to whatever is in that component, right? So we're going to have the V times X in the in the reactor itself divided by the QE leaving the reactor. This should go to 0 because XE should go to 0, right? So if our clarifier is working properly, that goes to 0. And then we have plus QW XW. I'm writing XW here. This is XW is XR, no difference there. So then what we're left with is theta C is V times X divided by QW XW. And so we notice then that the one piece that we have control over that can control the solid's retention time is QW. We can select how quickly we flow that. Now maybe we have some limitation, like we're producing so much solids that we have to waste something. But remember, we could have a very large recycle flow rate. We could take 99% of this and just send it back into the sludge tank, potentially, right? So we have a lot of control over QW. With a recycle system, we're generally going to have some amount of it recycle and some amount of it go to waste, right? So given that, that one piece ends up controlling our theta C. Or if we have just a set known theta C, we could notice that maybe this QW could change X itself. Okay, so there's one equation that we will use. So we've got two so far. A minute ago we looked at the F to M ratio. That's that food to mass. Now we have the solids retention time. And that's really, those two equations are, I think, about all we really need in order to solve problems with a recycle system. So you'll have those both on your equation sheet. Remember, these hopefully, obviously, do not apply to no recycle systems. Right, so we're making a big distinction between the recycle systems and the no recycle systems. All right, so we've got an example problem here. It's quite a multi-part problem. A couple of the pieces are practiced with the no recycle system, just like we did the example last time. So we're going to skip by those fairly quickly. But we'll go ahead and read through the problem itself and kind of touch on the pieces. I'll give you a couple minutes when we get to the point where we are going to kind of apply those pieces we just looked at to think about it and to solve it. But I'm going to try to go a little quicker than normal through this. Okay, so we're here to consider a wastewater treatment plant that's being renovated to include a return activated sludge, RAS line. Assume the influent MLVSS concentration is 0 without the recycle line, and the following parameters are known about the reactor which acts as a CSDR. So it gives us a bunch of parameters. It asks us to draw a schematic of the activated sludge system before and after the addition of a recycle line. Okay, so take a moment, sketch that up, and I will join you. Okay, so this should be pretty straightforward. Either case, we probably will be using a clarifier. In both cases, we probably have primary treatment, which would involve a clarifier up front. But whatever the case, what we're really after is just the secondary system right here. So really, in both cases, we're probably catching our microbes with a clarifier afterwards. That's pretty typical. Or having some sort of sedimentation system, but you could draw it this way. If you didn't draw the clarifier in the beforehand, that's probably all right. Maybe it's a stabilization lagoon or something. But we just needed a return line coming from the second clarifier. Okay, so then part B says, what is the final substrate concentration in the no recycle system? So it's looking for S. This calls back to the example we solved last time, where we did a mass balance on X, and we ended up with a formula for that. So I'm going to leave that to you to practice on your own if you wish. Part C, again, just like that prior problem, this is looking for the efficiency of BUD removal. So this is going to be something like S naught minus S over S naught times 100%. Likewise, D, again, following the same structure, is more practice. We can get there by solving a mass balance on S. Okay, so all of that is pretty much parallel to the previous problem. So if you wanted to walk through that to better understand it, there you have it. Part E says, and this is kind of applying what we did today, if the sludge produced has a concentration of 12,500 milligrams VSS per liter, so that's, I guess, giving us a new parameter there, which is now recycled with a recycle line at a flow rate of 150 cubic meters per day. What is the resulting, quote, influent MLVSS concentration in milligrams per liter of VSS entering the aeration tank? And I say, hint, refer to your drawing, so draw it up again if you needed, and do not overthink this problem. Okay, so take a couple minutes and approach that. I'll go ahead and write up the equations that we've done so far for this and the next slides, but take a look at that. Make sure to draw up your system to identify what it's talking about here. Okay, so hopefully you took a look at your drawing and recognized what we're really asking for is, well, we know that we actually have no significant amount of microbes coming in from the influent, right? But then we recycle, so what is the effective amount entering the system? That's what happens. What I'm asking for is in this portion of the drawing, right? What's happening there? And so we can find that if we do a mass balance at this juncture. So if we just draw a little control boundary around that juncture, it'll simply be what's going into that juncture is what's going out, right? So we've got Q times X naught will go to 0, plus QR times XR, what's coming in from the recycle line, is equal to what's in the total flow rate going in times the concentration of X going in. So I'm just labeling it X in, and we can say that that's going to be equal to QR XR divided by this Q total here, the Q in. So in order to find that, we can add, we can take the values that we know here, convert them all to the same units in terms of the flow rates. I think we've got cubic meters per second and then cubic meters per day, and get our answer that way. So let's see real quick. I might leave the calculations to you for later, but let's see if we can. QR recycle was, where did that go? 150 cubic meters per day. So it'd be 150 cubic meters per day times XR was 12,500 divided by the Q in. This is 0.01 cubic meters per second. We could multiply that by 86,400 seconds per day, and that should be 864 cubic meters per day then, instead of, after that conversion. So with that, we can get an X in value. Anybody have that for me real quick? Gonna make me do it? Okay. Is that right? Okay. Okay, so that should give us our X in value. And we can check, that should definitely be below the amount recycled. We are diluting it by adding it to the influence stream there. Okay, so then part F says, in the new system with the recycle line, well, let me just make a comment here. It would be pretty easy to convolute this, to complicate this problem if you're trying to use one of the formulas we covered without recognizing what the actual question was asking for, right? We really only needed a mass balance right on here. It didn't need anything fancy. It was just what's coming in is going out, right? But you did need to be able to draw the system to recognize that and see that for this piece of it. Okay, part F says, in the new system with the recycle line, the resulting food to mass ratio is 0.58 milligrams BOD per VSS per day. What is the new MLVSS concentration in milligrams per liter VSS? When it asks this, that's saying what is X? Remember, MLVSS, that's mixed liquor, volatile suspended solids, meaning that's in the system. take a minute and work on that. Okay, so this one should be pretty straightforward. We can take that food to mass ratio. If we know the food to mass ratio, we know the other parameters here, the flow rate, the influent substrate concentration, and the volume. We kind of have everything, so we just swap the variables here. Again, not doing anything fancy. This F to M ratio is just one variable here. So we're just swapping that with X, and we can put in our numbers here. Converting that flow rate that was in seconds to days, and putting in these other numbers. We can check the units should cancel for us well. So we need milligrams per liter VSS. We've got on the top milligrams BOD. On the bottom we've got milligrams BOD. We've got this milligrams VSS that should come to the top. The days should cancel, and the cubic meter should cancel. So everything should work out accordingly. And anybody have this number? Should be that right? Oh, I did something wrong. Should be 596. All right. Any questions there? Does that make sense? Okay. So part G says, what is the flow rate of the waste sludge line? So what is QW? We can use the, not worry about that part. So QW is the question. And here, if it was not already kind of obvious, we'll probably be using this theta C equation, which was the equation we had that dealt with QW. Okay, so we can just rearrange the theta c equation, recognize we've got theta c given to us in the text up here. I should have reminded you that the XW was 12,500. That was our amount recycled, or our concentration of the recycle. So when we put these numbers together, we can get a value for QW, and we'll notice that the units are going to come out nicely into cubic meters per day, given the setup here. So we'll take this. We'll say this is that number times 550 divided by 8 divided by 12,500. So QW should end up being about 3.3 cubic meters per day. Okay, makes sense? All right, so the last part of this problem here is just really asking, okay, what happens to the hydraulic retention time, like solve for it before and after. And this, it doesn't really matter. Effectively, theta is V over Q, right? That's not going to change before or after, given kind of what we learned about how we treat the nature of this reactor. Okay. All right. Any questions or issues? It kind of turns out we don't deal with so much the rates of bacterial growing in this sense, in terms of the recycle control sense. They're still certainly important. They're still, if you were to actually work in the industry or design a wastewater system, the rates at which these things are growing and that type of mass balance does matter. But from sort of a bit of a, when we go into that recycle system, we can take a step out of that depth of the mass balance in a way. All right, so now I'd like to turn to biological oxygen demand, since we were able to get through that pretty quickly. Just take me a moment. And we'll see how much of this we can go through. Okay, so we've introduced, yeah.
Is the new assignment tonight like 2 problems on one way recycle, one recycle?
Do I have 2 quizzes up or just one? Okay, I think one quiz is on the generic, neither of them are like calculation problems, I don't think. Or if it is, it would be very, very simple. I think one is on the recycle system and one is on the no recycle system. So it'll be like, one's gonna ask you about kind of, okay, what are the typical growth parameters type of thing? And one's gonna ask you, okay, well, which parameter that we just talked about controls the whole system, right? Like what's your lever that you can control with? Questions like those. Yeah, so please don't forget to take those as kind of participation assignments for you. All right, so we've already talked about BOD. We've introduced it as the microbe food, but we really didn't talk about how we measure it or kind of the mechanics of why we're putting microbe food in that term in the first place. And so that's this last section of our course is going to be about. It's really understanding oxygen demand. It's a conventional pollutant is that oxygen demanding substances. And it really is coming from any biodegradable organic matter in some sense. So it's going to be the defined formally as the oxygen in milligrams per liter demanded by microbial growth. And typically this will be, we will say BOD 5 is the convention. Probably talk about this in a moment. And that means it's that amount demanded over 5 days. the 5 is a bit arbitrary, but it does give us a pretty good confidence that the microbes had a chance to degrade stuff. So this is going to be, we can reference it or think about it in comparison to what we would theoretically, what we could theoretically demand in terms of oxygen as the theoretical oxygen demand, which would be a stoichiometric calculation. So if we took, for example, glucose, C6H12O6, we could calculate the maximum possible demand to convert all of that into CO2, right? So the theoretical would be to take, let's say, 1 glucose plus some amount of oxygen to yield, that'd be 6 CO2, and we'll probably have some amount of water. So if we were to check this, we've got that, let's go ahead and balance real quick. We've got 6 CO2. We'll balance the hydrogens. We've got 12 over here, so we'll need 6 waters. So then we have 18 oxygens on the right. We'll need 9 oxygens here. So we can see it's 9 moles of oxygen per 1 mole of glucose in this example. And you could calculate that in mass and say, okay, here's our theoretical maximum oxygen demand given this stuff. In reality, wastewater and natural water just have so much random molecules that there's no way we could do a theoretical calculation. And even if we could, in reality, some of these are going to form bacteria cells and not quite demand so much oxygen. And we're going to have some incomplete reactions. We're not doing a full, complete combustion reaction, right? Even if we were doing a combustion reaction, we might have incomplete combustion, right? So this theoretical is just that. It's theoretical. It may be a good reference point, and maybe in some cases you have let's say a sugar spill, or you're injecting molasses in order to stimulate bacterial growth. Maybe you get close to it, so it's perhaps worthwhile in some cases. But typically, what we would look at is what we're aiming to understand is that biological action. Overall, the goal is to understand the impact of our wastewater. So here's kind of the two extremes, biological and then theoretical. But we can actually also take a third type that's kind of in between and do a chemical approach, which could get us an answer a little faster. So if we look at the chemical oxygen demand, this is actually us taking a combustion type approach. Take our sample and run it through a system that should combust everything into CO2, and do what we can to just do oxidation. So we typically get more COD than BOD. So the chemical method of oxidizing stuff and demanding oxygen in that regard is typically larger than BOD, but theoretical is going to be the largest of them all. Okay, so COD, that's not talking necessarily about what it is that we're observing. It's just the method we're using to observe it, right? So we're using a chemical method to fry the stuff to see how much oxygen was taken. It's very similar to total organic carbon analysis, if you've had a chance to do that or look at that. In fact, it's nearly the same. So we're just going to give it high temperature, some sort of oxidizer, maybe a catalyst, sometimes UV. Just blast the thing and observe how much oxygen we're taking in order to yield CO2, water, nitrogen, whatever else. All right. So BOD, then, it's more accurate to what's going to happen in real life if we discharge some wastewater, but we generally do it on a 5-day type of experiment. So if we want something known about it today, then we go for COD, and that's our estimation. Okay, so then we can talk about different versions of BOD, specifically carbonaceous BOD and nitrogenous BOD. There are other types of subclasses maybe we could come up with But in general, these are the most important ones. Carbonaceous is kind of the generic, that's what most people think of when we think of BOD. Mostly it's carbon. So that's just sort of the generic form. We have some organic carbon. It's going to be consumed and eaten to result in some respiration, right? We're going to process that, breathe in oxygen, breathe out CO2. Then nitrogenous, BOD, this occurs when organisms can take, let's say, ammonia and convert it to nitrate or nitrite, or kind of go along that sequence, perhaps extracting some energy, perhaps, typically extracting some energy out of that, doing that conversion with a biological system. Or perhaps you're just looking at how much ammonia is actually being released during the processing of these, digestion of stuff. It's going to release ammonia, which then will go through this process. So overall, the net result is you're consuming oxygen during that conversion. And you'll notice here, this is actually not balanced, right? So this would be balanced if we said nitrite, and then we could go and maybe say two of these plus 2 oxygens would give us 2 nitrates. I think that's maybe just 1 oxygen, right? But regardless, the point is it's demanding oxygen, right? That's the point. So we could do a subclassification that way. It's good to recognize that the carbon-containing compounds are not the only thing happening there. But a lot of times when we talk about it, what we're first and foremost thinking is that the carbonaceous, or just kind of all of it combined and just see what happens in the bottle. Okay, so how do we measure it? Well, there's a few assumptions and principles we need to take care of. First of all, we're going to do our culturing. We're going to let bacteria consume whatever happens to be in the water. We're going to do it in a nice sealed bottle. We're going to do that so we can say we start with some amount of oxygen. That's just a known amount. And then we're going to let stuff grow, and we're going to monitor how much oxygen is still there. So we have a very controlled bottle. We're not going to let light come in, so we're actually not going to use a clear bottle like this. Instead, we're going to use an amber bottle that's going to prevent light that could induce photosynthesis. So if we get photosynthesis, we're creating oxygen, and that's just not what we meant to do. It's very typical to use these bottles about this size, though. That's why I kind of have this showing here. And you could use that bottle, just put foil over it, and wouldn't be a problem. So then you allow the natural biological activity to happen for 5 days, and you measure before and after the oxygen concentration. So you might even have a stopper that has the oxygen sensor built in, and you start it, you read it, you wait 5 days, and you read it again, right? Turn it on when you need to, or whatever. That's kind of the gist of it. That'll give you a number to say the oxygen went from some value to some other value. Now, we have a kind of an inherent limitation, because DO at saturation is not a very high number. That's usually somewhere between 8 and 10 milligrams per liter at room temperature. If you get colder water, you can get a bit higher. You're never really getting above 20 milligrams per liter. I think 18 might even be the upper limit for like water that's about to freeze. So given this situation, if you have a lot of BOD, you could reach 0 or below your detection limit on your system. And then the biological activity kind of has to just stop, right? You didn't actually give the microbes enough oxygen to consume the waste. So when I say here that the dilution is often required, that's because of exactly that. We have a limit of how much oxygen can be demanded in our little bottle before we say, well, we can't measure anymore, this experiment just stopped working, right? So if we recall that wastewater is typically, like low strength would be something like 50 to 60 milligrams BOD per liter up to something like 550, we recognize, yeah, we're going to have to dilute even the weak stuff in order for us to have enough oxygen in there to even make the observations we need to make. Okay, so that's a lot of what our calculations are about, is to say, all right, well, we're going to measure BOD before and after. That's our BOD. That's our BOD 5, right? If we wait 5 days, we could simply say, if we didn't have to dilute, then BOD 5 is just before minus after, right? It could just be 10 minus 5 equals 5 milligrams BOD, right? It could be as simple as that, but normally it's not. Normally we actually have to dilute, and so we're going to add in this dilution factor, P. We'll just call it P here. So if we divide that, whatever our answer was, by the dilution factor, that means if 1/10 of our little bottle was wastewater, P would be 0.1. So if we then had our number of 5 milligrams per liter, we multiply that by 10, or we divide by 0.1, and we find, okay, now we have a case where we have 50 milligrams BOD per liter. So if in our bottle we imagine that something like 10% is wastewater and the rest of it is DI or dilution water, that's kind of how we could imagine it, right? So if we fill this bottle to the top and we have it in that regard, we have some volume for the dilution and we have some volume for the wastewater. I guess we could just write a V there. So our P then is going to be the volume of the wastewater divided by the total volume, which is those two added together, right? So this would just be total. So it would often be something like, 10, or let's give it the 10%, like I was saying, 30 milliliters per 300 total. Something like that would be pretty typical, right? And that would be P equals 0.1. All right. Okay, that part's, I think, pretty straightforward. Any issues or questions there? Okay, so we can dilute the water. We can do our BOD 5. The point is to get our DO final. We have to keep this above 0, right? This must be measurable. That's kind of the point of the dilutions, right? We have to have a measurable amount of final oxygen. Okay, so a quick example then. We have a 10 milliliter sample of sewage mixed with enough water to fill a 300 milliliter bottle. Has an initial DO of 9 milligrams per liter. And to help assure an accurate test, we want to have at least 2 milligrams per liter of a drop from day 0 to day 5. So we want at least 2 milligrams per liter change during the 5-day run, and the final should be at least 2 milligrams per liter. So kind of what we're saying to ourselves is, like, we know we started at 9. We want to make sure that it drops enough so that we're confident that our instrument's not just a little bit variable. And we want to leave enough DO, so, again, because we're not so sure about the instrument's accuracy, perhaps at below some level. And then the question says, what range of BOD 5 could this dilution produce? For what range of BOD 5 would this particular dilution produce the desired result? So take a couple minutes and look at that. I sneeze. I'll be right back. All right, so we can look at this probably a couple of ways, but I think if we consider what's the minimum BOD, to look at like that range of BODs, right? We're looking for the min to max that could get us a reasonable result here. The minimum would be the case where we recall the equation itself that we're looking for or that we're using is BOD is going to be equal to the DO initial minus the DO final divided by some dilution factor. In this case, we can find the dilution factor. We said 10 mils of sample sewage and then mixed with enough water to fill a total bottle of 300 milliliters, right? So 1/30 is our dilution factor. Okay, so then for our system here, the minimum BOD, that's going to be the least impact. We were told that we have to have at least an impact of at least a change of 2 milligrams per liter, right? So in this case, we know our initial is 9, and well, we could write this one of two ways, right? We could say whatever up here leaves us with 2 is our minimum. So 9 minus 7 divided by 1/30. Or we could also write it as, well, the minimum drop acceptable is our minimum BOD value. So we could just write that's, from the get-go, our minimum drop at the top, recognizing that that's 9 minus 7. Okay, so we could, in either case, do it that way. And if we go over to our calculator, we'll say that should really just be, is that 2 times 30, essentially? Don't need a calculator. So that should be 60 milligrams BOD per liter. And then the maximum would be when we had all but 2 go away. So the drop, in that sense, would be 9 minus a final value of 2 over 1/30. So that would be like saying 7 over 1/30, and we could say that should be 210 milligrams BOD per liter. Does that make sense? Okay, so we're going to complicate it just a little bit and say, well, when we do this dilution, it turns out that if we use a whole bunch of really dilute water, we're not going to have enough microbes hanging around anymore sometimes. So we actually have to dilute it with special seeded water in order to give the system some microbes to work with. It also turns out that when we need to add bacteria, well, bacteria we typically like to keep them in an environment where they're able to grow and survive. So we typically have a little bit of biological media in that, meaning when we seed the water with some bacteria, we are also seeding it with a little bit of BOD. So practically speaking, it's not a huge complication, but it's kind of just an annoyance type of complication. We are adding a bit of BOD, and so we have to back calculate to say, okay, well, let's make sure we counteract for that or account for that when we do our final calculation. So if we are to look at our bottle of wastewater again, maybe we have some amount of wastewater here, and then we decide, well, we also need some seed water. So we could look at it as if we have a little bit of seed material there, or we could look at it as in the entire rest of it is just the seed water in general. Normally, instead of having pure dilution water, what we'll just say is the rest of this to the top is this. It's not like pure water. It's still our V dilution, but it's got that, it's got more stuff in it. Okay, so then when we do our calculations, we have to account for 2 BODs. And typically the way we'll do this is we'll simply set out 2 bottles for every 1 experiment. We'll take the seed water just by itself and say, all right, well, we're going to spend 5 days anyway. Let's just set up a bottle so that we know how much BOD was in our seed water. And it's not going to be a ton, so that we don't usually need to dilute, or it'll be a small dilution if we do. So we'll have just our seeded dilution water sitting in one jar and our actual experiment in the other jar. And we will say that our total BOD5 of the mixture is going to be, times the volume of the mixture will be equal to the BUD in the wastewater times the volume of wastewater plus the BUD in the dilution water times the volume dilution, right? So all of that to say, we have a mass balance on BUD now with 2 sources of it in our water. If we can find in those 5 days this value, we can know that, then we'll calculate the other part, right? So like I said, it's just kind of an inconvenience, complicates the calculations a little bit, but it's not a big deal, right? Okay, so M stands for mixture. I already have it written there, so just erase this nonsense. M is mixture, D is dilution. So one thing we can note, by the way, is if our P is the volume of the wastewater per total volume, 1 minus P would be the volume of the dilution water divided by the mixture, the volume of the mixture. So that can be handy, and we'll see in a moment that can go into our broader calculation and simplify some things. So if we want to solve for just BUD of the wastewater, this is just going to be a rearrangement of the mass balance here. We have this, the mass balance as the mixture, BUD in the mixture volume is equal to the other things. But if we just wanted to rearrange and solve for BUD of the wastewater, we can do that. We can go ahead and subtract some things from both sides and then divide by the volume of the wastewater. So that's why we'll see BUD of the mixture over here times the volume of the mixture, and then dividing by the volume of the wastewater. So this is just taking a couple steps in one here, subtracting this from both sides, and then dividing everything by this, okay? So then we have minus this BUD in the dilution times that volume of dilution divided by that volume of wastewater. So here's where this guy can come and simplify some things for us. We can say the BUD of the wastewater is then equal to the BUD of the mixture times 1 over P minus BUD in the dilution water times 1 minus P divided by P. Do all that and it can simplify down to finally this value here, BUD in the wastewater is equal to the BUD in the mixture minus the BUD in the dilution water times 1 minus P, all that divided by P. Okay, so we can sort it out to the point where the volumes all are arranged according to just the dilution ratio instead of having to pick apart each actual little volume there. So that's pretty convenient, and you will have this equation on your formula sheet as well. So just wanted to show we can get a value specifically for BUD of our wastewater given all that. Okay, so I think this is a good spot to leave off. We just have some practice and then we get straight into the kind of the modeling portion. So we'll leave off here, and after Thanksgiving we will pick up here. All right, so have a happy Thanksgiving and safe travels if you're traveling, and enjoy some good food. See you next week.